So I'm going to talk about Ncardia and also about Celistic, a company that we launched yesterday. So Ncardia is an iPSC stem cell platform technology established in 2014. We are approximately 50 FTEs right now, working in fact on drug discovery and on cell therapy. Growing towards 80 at the end of the year. Team is very focused on manufacturing of iPSC-derived cells, assays with those cells, screening, and assay development. So we are in Europe, in Leiden, south of Amsterdam, and in Gosselies in Belgium, south of Brussels, and expanding in the US. So company is really focused on this fundamental question of better cell technology. So we started on the left-hand side in drug discovery, recognizing that therapeutic developers always face this funnel where lots of compounds go on top, only a few enter clinical trials, and especially with advanced therapeutics. Human biology is not an option anymore, but typically a must. So what we have been doing a lot is building the best human cellular models, also genetic models, to really test whether therapeutics are effective and safe. Then on the right-hand side, we actually see that CAR-T therapies are hugely successful, but it's still very process-centric, so large manufacturing cost, which reduces access to patients. And so we basically see that radical new cell technologies are needed to actually get this product-centric again. So what we have been focusing on with Ncardia is mostly on the left-hand side of the slide. So starting with humans, which can be patients, we are making those iPS cells, and we focused a lot of our attention on getting to what the right cell in a scalable, reproducible, consistent way. Because we needed a lot of cells to actually use those cells at the earliest stages of drug discovery in high-throughput screening to be able to ask fundamental questions like, in this library of molecules, is there a compound that can reverse a disease phenotype? So today we are working with therapeutic developers across gene therapy, small molecules, biologics, RNA therapeutics, to answer those fundamental questions. Is my therapy changing the disease phenotype fully based on a biological system? So what we learned there is to manufacture those cells at scale and to deeply characterize those cells. And if you take 2 steps back, then those are actually the capabilities that you need in cell therapy as well. You want to get towards the right cell where you're confident of infusing those in a patient. You want to characterize those cells deeply. And we have actually seen in our first cell therapy projects that every single cardiac assay that we have ever developed on the left-hand side got used on the right-hand side. So there's a real synergy there from a technical perspective. From a commercial perspective, it's actually a little bit different. So what we are doing today is using our established portfolio of cardiovascular and CNS cells in drug discovery projects under the brand name Ncardia. We have successfully completed 60 of those projects. We'll do a lot more. This company is in hyper-growth mode, growing 100% this year and 100% next year. So we did our first cell therapy project with Blue Rock Therapeutics. They licensed our cardiac manufacturing technology now 2 years ago. And following that success, we actually said, let's continue to focus on immune cells. So on the cell therapy side, we are working on microglia, monocytes, on macrophages, on T cells, NK cells, where we really want to have that whole portfolio available for cell therapy developers to take advantage of. One day, we're actually going to bring those cells back in the drug discovery business as well to again, look for that synergy. But for the rest of this presentation, I will talk about Celistic, which is now 1 day old, launched yesterday. So what we want to do with Celistic is to create a path for therapeutic developers to find the future of cell therapy. And what we are going to do is to put all the processes in place, from reprogramming to gene editing to manufacturing, to work with therapeutic developers, which can in fact be autologous developers, to help them to transition towards iPSC technology. So it's a dedicated service company, but not competing on square meters GMP facility. It's competing on technology that we actually bring to the table. So the goal, of course, is to change the game in cell therapy. We all know what we call cell therapy 1.0, 2.0, where the cell source is patient-derived or donor-derived, where genetic engineering is typically done with viruses and per definition random and variable. The characterization, because it's so process-centric, is imprecise because there's just no time to do it in another way. Leading to a somewhat heterogeneous product where manufacturing is limited by limited dose availability and the cost per dose is incredibly high. So iPSC cell technology has the potential to change this because we would start from a renewable master cell line. We would do that editing once in that master cell bank and not all over again, which allows you to characterize that cell very, very deeply. Gaining you a homogeneous product that is becoming off-the-shelf available at a much lower cost per dose, and a dosing that can be done with multiple doses and multiple dose cycles because the product will be cryopreserved. So lots of benefits for the patients and for the therapeutic developer if you get it right, but this is complex biology. So the key benefit, of course, is to offer economies of scale. We are going away from one batch per patient to scale up where one batch is serving many patients, where you get rid of donor-to-donor comparability testing, where you can do your edits once, where you get economies of scale due to raw materials and labor. You spread your costing test over multiple doses, again reducing those COGS, reducing labor, and then gaining greater capacity. I think the fundamental problem in this field is that not a lot of iPSC developers have actually touched a bioreactor. In fact, 99 of the publications will talk about a 96 well plate, and I think that's the biggest gap that the industry is facing right now. So what we have been doing the last 10 years, and for me personally the last 18 years, is to play with that complex biology, which is in fact a lot more complex than what biotherapeutic developers are seeing today. Because think about you starting with a stem cell that can become all 220 cell types in the body. So controlling that and at the same time gaining scale is really the next level, probably in order of magnitude more complex than a gene therapy or an autologous cell therapy. And so this is the value proposition that we are bringing. We have been playing with this a long, long time to develop bioreactor-based manufacturing platforms focused on yield, on phenotype, on functionality, well-characterized cells that are potent at a lower cost of goods. So one of the key lessons that we have learned is that CMC actually starts at day zero in this field. So playing around with small dishes is actually not going to give you a process. We have actually seen over the years, over and over again, working in a small dish, going to the next dish will actually mess up your differentiation. So it doesn't work. These technology limits are very steep. And so basically you have to redevelop your process when you go to the next technology. So what we have learned in that process is that stir-tank bioreactors actually might also have an S-curve, so an end where you can't scale further. But in our experience, we haven't seen that. So processes that we have developed at very small scale, at milliliter scale, we have successfully 1,000-fold scale to liter scale, getting to billions and billions of cells. So an example of that is the work on cardiomyocytes that we have licensed to Blue Rock. That is now a very well-defined process, even without any cytokines. It's purely based on well-defined components, small molecules that basically drive those stem cells first to mesoderm, then to cardiac mesoderm, then to cardiomyocytes. And what we are routinely getting for the last 3 years are typical yields of 3 billion cells per liter, continuously producing batches of 20 billion cells in a 6-liter bioreactor. And so what we have seen in that process is, so this is a plotting of cost. So first we did it in hyperstack. So that's actually the actual production cost on materials, on labor, on facilities. So to just give you a little bit of flavor, so we produced with a team of 10 people 20 billion cells in a year in a facility. In the second year, we did it a little bit better, got to 20 billion cells with that same team. Then when we completely redesigned that process in bioreactors, We actually cut down on materials, on labor, on facilities, and actually a team of 2 could actually produce 100 billion of cells, whereas in a month, whereas we did a lot less in the hyperstack process. So approximately a 16-fold reduction of cost and a 50-fold increase in yield per FTE, thanks to complete process redesign from scratch. So our partnering model right now is that we are working really, really hard on those manufacturing processes for the immune cells that we're interested in. We are interested and working with partners that have ambitions to go in the iPSC space, that typically have gene edits that they want to have in the cells. So we will combine that, work with therapeutic developers to create a candidate product. And what is of interest for us is the manufacturing rights for clinical and commercial manufacturing. So we are a pure service company, no other therapeutic products. There to work with therapeutic developers to get these therapies quicker to patients. So on our GMP manufacturing focus, we are working on facilities in Europe and the US. What we will do is everything from reprogramming to gene editing, to creating the master cell banks, to creating the differentiated cells, cryopreserved and ready for clinical trials. And with that, I'm going to end the presentation. I think I've room for 1 or 2 questions.